I'm just going to start, and feel free to interrupt me at any point if you have questions. Okay. But basically, where we are right now is we have a unified image model for all the different types of image data. So if you had interacted with the site months ago, you may have been familiar with a different model where we had one section for H&E images, one section for immunofluorescence, and another section for immunohistochemistry, which confused everyone, another section for in situ hybridization. We've combined that all right now. So we have basically one place where all of the images are. And they're categorized differently. You can search on the different image types, but it's basically sort of one-stop shopping for images. So these images, there's some core data that's always going to be present for each image. You know, what species it comes from, the stage or age, the fact that there's an actual image file. Those are going to be the same for all of the images. But then different image types will have optional data elements. If antibodies or probes were used, if expression regions were scored, and so on. So I'm going to start.
Before we move on, I'd like to also emphasize that, you know, even though we focus the data type on imaging data, however, specimens is involved in most of the data submission.
Right, but what we're talking about now, I mean, this is the imaging tutorial.
Okay.
Okay, so
I just want to make sure that we are on the same page. So how many people are planning on submitting imaging data?
I guess that's a question for everyone but me here.
Well, potentially in the future. So.
Okay.
Yeah, we are going to have that kind of data. So.
We'll also have data.
Okay. All right, so I think that sounds good. So yeah, so we focus this tutorial on submitting imaging data, but the specimen, the same process of submitting specimen is also used for other data type as well. So it's just good to know generally that this is actually a crucial kind of information that we use in our repository.
Right. I mean, so the thing about specimens is that this is a core set of data information that is used for different data types. So I think that no one's ever going to decide they want to submit a specimen. They're going to decide they want to submit some image data or some RNA-seq data. And as part of that process, they will be filling in some specimen information. But if we could get back to the imaging here, there are sort of three basic models of how you might have images. So one is a really simple thing. You have one specimen, you have like one biosample that you put on a slide, you take one picture of it, and this is, you know, you have your one specimen with your one image associated with it. So another possibility is that you have, again, one biosample, and you take, you know, several different images of it. And then the third is that you have a biosample and you subdivide it into different sections, and you take some images of those, but you want to maintain this relationship that everything came from this one individual specimen. And so we support all of these different models. Okay, so the very first thing you do is create the specimen and then add images. And if you are doing this third model where you have the parents and children, you create the parent first and then add the children and the children's images as above. So I'm just going to show you this. And this is on our staging server, which means that none of this data is real. Okay, so I've chosen create specimen from the create menu. We have several fields here. This internal ID is something that is optional that you can fill in to help you locate your image. With some of our offline tools, it's useful to have this to link what's in the catalog with what you're doing offline. So it's convenient. I'm just going to call this Laura test. You don't need to always, but it's just a good idea to do that. There are some required fields. Species is one. Stage is technically, you can create something without a stage, but we want to have the stage in there. And if you don't have staging information, you can type in the chronological age, the number of days. Assay type is what kind of specimen it is. So for images, we have histology, immunohistochemistry, and in situ hybridization. I'm going to select IHC here. I'm going to select IHC here. Preparation and fixation are basically, what the slide is. And I'm choosing randomly because I don't actually, I'm not actually a biologist and I don't know any of this. So again, any of this information that you have, it's useful to fill in. So these are not required fields, but if you know if it's wild type or non-wild type, which presumably you do, if you have a phenotype, and if you know the sex, all of these things are good to fill in. If this comes from one of our cell lines, you can select that. If not, just leave that blank. If you have any notes, you can type those in. The parent is what I was talking about before when I said that you can have this relationship if you subdivide a sample. The representative image will come up later once you've uploaded some images. You can choose one to represent this specimen in displays that use thumbnails to kind of show a specimen, like you might be familiar with our gene page display. The PI, you select your own PI. And since this is just not real data, I'm going to choose our UX consultant as our PI. And then whichever consortium it is. So once you've filled out all these things, you can click submit and you have your new specimen record. Now, if you want to create children, which most of the time you won't, but again, if you've subdivided it, an easy way to do that is to hit copy, because this will fill in all of the information from the previous one. And the reason that that's convenient is that probably things like the species and stage and sex are going to be the same. I'm not actually going to do this. I'm going to
hold on. Can you also demonstrate if you want?
Yeah. If you want to create multiple ones, then you can also just hit this plus sign as many times as you want, and it will copy what was in the previous one. But those are just shortcuts. Okay, so once you have created this base record, one other piece of information that we want to add is an anatomical source. And that's basically the sort of gross anatomy of what's on the slide. So it's not a very fine-grained thing. Usually it's going to be something like, kidney. Add one of these. And so we'll choose menanephros. And so that shows up here. And that's a bad example because it's not consistent with our stage, but that is the basic process of creating the specimen. What we don't have yet is the image that we're associating with it. So we'll create one of those. And let's get a happy, I'll show you that you can create more than one at a time. So what we do basically here is select a file for the image, and then another for the thumbnail. And then we have to choose again. Did I do this badly? If I do the plus here, will it copy more?
Yes.
Yeah. So, all right. So I clicked way too many times, but you can see now that all of these things that I had selected show up as additional values. Okay, actually I was going to create a second one. Sorry.
Also, if you want to create, let's say 10, and without wanting to click, the plus 10 times, if you actually click on the arrow down button, then you can actually specify the number of records that you want to duplicate.
Okay, so now that I've filled all these things in, I'm going to submit. And we now see that we have these two images. So if we go back to the specimen, we have a couple of things we can do. One is we see the images there. And the other thing is that we can select one of them to represent the specimen. So these are the basics. One other thing that I wanted to mention about specimens is that this doesn't make sense for this one, but if you have something that is, say, to stack with multiple channels, you can actually add image channel information. So you can say, for channel 1, the color is red, and you can label what it is. But that doesn't make sense for a JPEG. Okay, so any questions before we go back to the presentation?
Yeah, Laura, I was just wondering, with organoids, where would you put the age of the organoid? Would that be under chronological age?
Yeah, so that is something that we're working out with Todd, and I don't think that we actually have an answer to that yet. It's a good question.
Right. So for the age, I think we're going to use the chronological age because the stage is not sufficient to address that issue. I think so far we advised you in the past to use the chronological age, so that's still hold. However, if you want to actually add information about where the organoid line is from, I think we still try to standardize on that.
Yeah, I saw there was somewhere that there was a drop-down selection for line, so that's okay. It was just the age, but that makes sense to use chronological age.
Yeah, I think there's also a question about the anatomy, how you specify that it's an organoid when you're choosing the anatomical region.
And none of those options, it didn't look like they had an asterisk, so they're not essential. Is that right?
Yes.
Like for example, anatomy, you don't have to be able to submit them.
The anatomy, we really, really want people to have it there, but I don't think we have a solution for organoids at this point.
Okay.
Yeah.
So one thing that I'd like to point out, if Laura, you can go back to the specimen page, is there's basically a hint. It's actually the record status in terms of what's missing, and so if it will give you a hint in terms of what we expect for the record to be complete. It's really minimal right now, but usually it's expected for you to have either this the tissues anatomical source or the cell line. So if you don't have it, it will complain, and then that's supposed to help you guide you through the whole data submission process. So,
yeah, so if we remove that, you can see this here. The other thing is that since we called this an image type, it also complained before we added the images, because if it's an image type, it expects there to be at least one image. Okay, and then some hints. If you upload a CZI file, we will process it so that you can see it online in our viewer, but that happens asynchronously, so you won't see the nice viewer view right away. Another thing is that you can also add videos, which people do sometimes if they have 3D sections. And I'm not going to go through that, but it's almost exactly the same as the adding an image process. Okay, so at this point, you have some basic stuff in there. You have all the information about where the image came from, or the sample that the image came from, and you have the image itself. But then there's more things that you can do depending on what type of imaging data it is. So if you have used an antibody, then what we would like you to do is tell us about that. So here's just kind of a sense of how our model treats antibodies. So use an antibody to probe for a protein. And to link this to a specimen, we have a type of record that you create called specimen antibody. And that tells you a couple of things. It tells you that you've used that antibody on that specimen, and it also lets you specify additional information like the dilution, any secondary antibodies that you used. So more information so that people can interpret the image. And then once you have created this link, the protein that was probed for on the antibody will appear on the specimen page because there's a path to it. And so again, switching back. So we're going to add an antibody. So we already have a number of antibodies in our system. There are, if you use one that's not in here, you can actually add an antibody record, which is pretty straightforward. For this, you just fill out the information in the form and submit it. So I'm not going to go through that, and I'm just going to select these rabbit antibodies. And again, you can add the dilution. And lab ID is just if you have an internal ID in your lab that you use for the antibody, you can type that in here and submit. And so now if we go back to the specimen page and we look up here, we see we have this protein ACPP, which has shown up because we've added an antibody that detects ACPP. So those are antibodies. And going back to PowerPoint. Okay, so similarly, if you've used a probe to probe for a gene, again, probes are linked to genes because that's what they look for. We have this table called specimen probe that will link a specimen to a probe. The probes we have many, many probes that are already in the database. You can add more if you have a probe that's not already in there. And this is very similar to the antibody process. So again, I'm just going to show one. And I'm just going to use an existing probe. But again, and so all of these fields are optional. You can fill them in if you want. I am not going to bother.
But we recommend that you fill in as many as possible because they actually involve in the search. So that will make your records more searchable.
Yeah, I think some of these are not necessarily involved in the search, but they're useful for they're useful for knowing how the experiment was run. And then since we've added this probe, we see the gene associated with that probe has shown up in the record. But yeah, I mean, in general, we kind of encourage you to add as much information as you have. Okay, so at this point, we have this kind of weird specimen that we've created that has a little bit of everything in it. But we're going to see now if you've done expression scoring, so you've added either a probe or an antibody. So you're looking for, the slide is showing either a protein or a gene. If you've looked at it and scored fine-grained anatomy to say whether or not you found it, then we'd like you to add that information as well. And so going back, you can look at this and say, okay, this is a very weird thing because we're looking at the metanephros and our fine-grained thing is the angle joint, but whatever. So you can do this as many times as you want. We care about whether something was not detected as well as whether it was present. So any of these values, so we'll say not detected here. You can add additional things about whether it was strong or weak. And these are basically the same kind of values that were in GUDMAP 2 if you're familiar with those. We have these patterns that you can specify, pattern location, whether it's, well, you can see these values. Density change. So again, as much information as you have, but definitely the place and whether it was detected or not are useful things. Okay, so I should actually choose something that's valid for that stage. I'm trying to remember what the stage was. Sorry. So we have a couple of views of this, so you can see this either as a table or as this nice tree view. But I have somewhat foolishly added something that's not valid for the age that we have, so the tree view doesn't particularly like that. So I'm going to show you something that actually looks good. Okay, so now when we go back to the specimen, we see this and it's nicely highlighted. Okay, going back to the presentation, I think that's actually the last thing that I added in terms of adding data. The final steps are to use the curation status. Are you guys familiar with this from, oops, are you guys familiar with, sorry. Are you familiar with this from other data entry, or do I need to go over the curation status? Okay, since I'm not hearing lots of people saying, yes, I know this already, we have a workflow for curation status. Everything starts out in preparation. When you have something finished, to the best of your knowledge, you can change it to submitted, and then the system will decide sort of where to dispatch it. And for specimen data, it gets dispatched to our biocurator. So you'll fill out submitted, it'll go into biocurator review. It's still not visible to anonymous users. It will only be visible to them when it's released. And so our biocurator will either release it so that everyone can see it, or he'll get back to you and say, ask you some questions and see if there's anything that needs to be refined or added in. But, you change the curation status.
Sorry, Laura, I just saw there that it said specimen something incomplete because it needed an anatomical source. So will it not, even though it doesn't have a red asterisk next to it, it's still not going to let you submit because you're missing some information? Is that right?
No, actually that's not right. Although I can see why you would think that. This record so the curation status is something that is meaningful to the system, and it won't, the system won't show it to anonymous users unless it's been released. The record status and record status detail are set by the system, but they're more advisory. So if there's a reason why there's no anatomical source, then that's fine. And so that's kind of between you and the biocurator.
Okay, so it's just letting you know, but it will still submit it.
Yes.
Okay.
Yeah.
So just to answer your questions, whenever you see the stars on the page, that means it's required and therefore you have to fill it in. If not, then it will not allow you to submit. However, when we look at the record status, it's basically look at more than just one record. It look at the main specimen and also things that it kind of thing that is involved in this kind of assay type. So right now the record status is really simplistic. So it's only checked for an anatomical source, check to make sure that either you have an age or you have a stage in the record. But eventually we can expand that to be a little bit more thorough so that it can guide you a little bit better. But right now it's really simplistic. And like Laura was saying, it's meant to be supervisory. So guide you in terms of what we think is needed. And it can be adjusted if you feel like it's actually reporting the wrong thing. So let us know if it's actually not doing its job.
Yeah, and the other thing we should mention is that this the record status is only visible to, and the record status detail are only visible to people within the project. So outside users who come in and look at it won't see this big incomplete flag. Okay, and I should mention one other thing. Depending on how things work in your lab, if you have someone who is not the PI, who is working on things and they want the PI to look at it before submitting it, they can also set it to PI review. And then the PI can come along and set it to submitted. But I'm just going to click submitted here. And then we'll see that this has magically changed to biocurator review, which means that Todd will look at it and either release it or talk to you. And then the final thing is if you have a collection that you want this to be part of, you can add that to the collection now, actually at any point after you've created the record. And that would basically be the final step.
Laura, can you explain a little bit more of the table of contents? So what are those options that are available there? And then how do I know what to pay attention and what not to pay attention?
Right. So this is a little, so we have one table of contents that is used for everyone. And so it's used both by people who are browsing the data and people who are entering the data. And what the table of contents shows you is all of the information that's not directly in this record, which includes things like the gene and the protein, which kind of look like they're in the record, but they're actually in associated tables. So someone who is browsing for things would probably want to find the gene pretty quickly or find the protein pretty quickly. As you're entering information, you wouldn't be that interested in doing this because you're not entering those. Those are getting entered for you when you enter your antibodies or your probes. So this table of contents, anatomical source, it's a little redundant to have the table of contents for the things that are actually in line, but that's how our systems work.
So I guess
Where
what
it's mostly useful is for things that show up way on the bottom, like the images, the video, fried specimens. It's just a way of, not having to scroll down to all of these individual faces. In terms of what's important, I think you basically know what's important by what you've done with the actual specimen itself. So you would know that if you've used an antibody, you want to add an antibody record. If you've used a probe, you want to add a probe record. If you've done expression, you want to add expression records. Expression scoring, you'd want to add expression records. Is there something else you wanted to say, Honsuda?
Yes. So basically, I guess what I'm trying to suggest is that it would be very helpful for you to go over the table of content and then see basically which relevant information that you think should be added associated with your specimen. For example, there's basic acknowledgement, right? So this is minor, but if you want to acknowledge somebody, there's also a place for basically put information about publication that's involved in the specimen, and there's a way for you to add the notes, the results, so things like that. Even though they're minor, they're there. And so if you go over the table of content, it will tell you in terms of like, what are the related information you can add to the specimen. And I think that's usually very useful.
Yeah, so you can do that through the table of contents. You can also scroll down if you're not sure what some of these things are. I think some of the column headings might or might not make that easier to see. Are there any questions? Everyone's really quiet. I do have links to more information, and these slides in this video will be available on our CRISPR World AV.
So Laura, can you do one more demonstration to a common case that actually keeps happening in the case that when you need to create multiple specimens with the same anatomy? What would be the kind of like trick to do this in bulk?
I don't know the trick to do this in bulk. You can walk me through that.
Okay, so basically you would first create multiple specimens, right? Let's say you create.
So should I like copy this one?
Right, and then let's say you create 3 of them, and right, and then you might have to change your internal ID, right, Laura, or internal ID.
because they have to be unique.
Okay.
And then click submit. Then you will have, this many number of records created. And if you go to any individual records, it will complain that specimen, it will complain that you don't have an anatomical source. To actually add the same anatomical source to multiple specimens, what you can do is
I see what you're doing.
So you can either do the trivial way by going to each specimen and add one at a time, right? Or the trick is when you link the data together, it can go to any direction. So the trick is to go to the specimen. I'm sorry, to the anatomy. So the method that Laura just showed is actually straightforward, but you need to add one at a time, right? So if you want to add the same anatomy to multiple specimens, then you can go to the search. No, you have to go to the search. You're going to have to go to the search menu and then click specimen. No, I'm sorry, anatomy.
So anatomy terms, anatomy tree view, or faceted search, either way.
So let's go for the preview for the one. Okay, see what we did.
Mm-hmm. And then click one of them, the one that you are going to associate with the specimen. And then you can actually add the specimen to the terms.
Not sure what.
Yeah, staging's a little slow.
Right, so if I type my internal ID or the prefix of my internal ID here, I can limit it to these ones that I just created. And then
Right. You can click all. Right.
And then if I look at any of these, I can again see that it's there.
So this is actually a good trick, for when you have to do the same kind of similar specimen in bulk, which just save you some time.
Cool. And then as far as finding the materials from today, Laura, can you demonstrate, like if you go into the help menu, how you could get to the documentation?
And
Emitting
then
data. And then scroll down and look for specimen. And you click that, and it's not there now, but at the top of the page, there'll be some links to the slides and to the webinar. So we'll post those later today. And I'll send out a direct link to this to the list as well.
Any questions or feedback?
No, that all makes sense to me. Thanks, guys.
Great.
Yeah, it was very helpful. Thank you.
Awesome.
Yeah, thanks very much.
Sure. And then, of course, if you have any questions, help at GUDMAP.org or rebuildingthekidney.org. They both go to us.
Laura.
Actually, one more thing, Laura, you forgot to show the dashboard. That one is basically a good summary of what you have submitted, and if you want to go directly, to the your submission.
Right.
Nice.
But I think somebody did have a question. Was that Manoj?
Yeah,
Or- yeah.
yeah. So I know this is mainly this session was specific for image data, but we have to submit some RNA-seq data, and is there a link or something for how can we submit?
Yeah, we have done previous tutorials on RNA-seq data. So if you go to the submitting data page that I got to from the help menu, and you click on sequencing data over on the right, we have basically some written documentation. We have slides from a previous tutorial, and we actually have the webinar from we have the video from the previous tutorial.
So
Okay,
sequence
thank you.
data
Yeah, I mean, my PI just told me today, just try to see this. Maybe it will be helpful. But anyway, thank you. I'll go through this sequencing data tutorial. Thank you.
Great. Okay.
Okay, Laura, can you show the dashboard just a quick?
Oh, right.
In case it's convenient to people to submit a lot of data.
I call this GUDMAP or RBK. I call it GUDMAP. And so, yeah, in fact, since this is the most recent stuff that was submitted, and Cia Romano is not a real PI, so this is the only data from her. We can see that we created a couple of images. They're complete, but they're still in preparation.
Right.
Yeah.
And if you click on a number, it will direct you to those records.
So how come everything in there says GUDMAP?
Oh, because that's the GUDMAP dashboard. So if we want
Plus, this
to
is staging, so we've just been adding stuff in there too.
No, but this is the RBK dashboard, so.
Oh, right, yeah.
Yeah.
You can toggle.
So that's
Okay, I
based
see.
on. That's
You've
based
got it
on.
clicked on the left there. Okay.
Yeah. So that's actually based on the consortium field in the record that you fill in when you create the record.
Right, that makes sense.
That's it.
Okay, great. Thanks everybody for coming out, and we'll be sending another email later with links to today's session. And have a great day, and we'll be in touch.
Thanks, Chris and
Okay.
Laura.
Thanks, guys.
Thanks.
Thanks. Bye.